experience this interaction with the tumor. And the claim of the authors is that if we can understand this at systemic level, then we can design better immunotherapies. And the main question here is that, and the main question of the paper, as I could interpret it, is how does tumor experience affect microenvironment of the immune system? So the technique that they use to solve this question is mass cytometry. So in contrast to flow cytometry that a lot of us know already, flow cytometry is limited to the spectral overlap. So you can use several colors, but there's a point in which the spectral overlap won't allow you to go further to use several markers to do a phenotypic classification of your cells. In that sense, if we use mass cytometry, instead of using spectral colors, what we use is mass. And the way we can use the mass allows us to use much more combinations of isotopes to mark the cells. We can use much more combinations that will allow us to do a really high throughput cell phenotyping, in which we can use several markers at the same time and then pull all the cells at the same time to do really high throughput experiments. The way this works, this technique works basically with a very similar principle to flow cytometry, in which you have the cells and then you use, as in cells and as in classical flow cytometry, you would use antibodies. And these antibodies, instead of being coupled with a fluorophore, the antibodies will be coupled with a metal ion, with ions, will be coupled with metal modifications. These modifications to the antibody itself will be then passed through like a single drop, and these drops will be ionized. The ionization of this will generate something that is called like the ion clouds, and these ion clouds will be then filtered to remove like impurities, and the interaction, like the modifications that were conjugated to the antibody and the metal ions, will be then quantified by mass spectrometry. So this spectrometry will give us a mass, a known mass that we know because we conjugated the antibodies, and then this will let us to have this filing, which we have a cell, and then the different type of markers that we use, and the enrichment of the different markers that we use in the whole pool of experiments. So in this case, instead of having a unidimensional data, we have a multidimensional data that we can then do a more complex analysis in order to classify these cells based on the different types of markers, but even better to classify each sample by all different types of cells that compose this pool of each experiment. So the main findings of the paper that we are going to discuss today are that systemic immune organization is altered across multiple tumor types. We are going to discuss how immune cell states are dynamically altered across immune organs with tumor growth, and how the novel T cell responses are impaired in persistent malignancies, and how tumor resection reserves in systemic immune organization and responsiveness. So the first result that we are going to discuss is about the systemic immune organization and how it's affected across multiple tumor models. So far, I don't know if the authors would like to add something regarding like your research question or the importance of some of the approaches that you took.
Hi, this is Kamir. This is like a really great introduction. I guess just emphasizing that almost the entire field is focused on the tumor microenvironment. And there's been also we've been kind of isolated in thinking about the macroenvironment until really this year. Like a couple papers came out in Nature Cancer, an interesting paper in Nature Medicine last year that are continuing to build on the idea that we need immune cells that are either generated from the periphery or invade the tumor from the periphery. And it's not just an isolated system, you know, we need everything. So this is really the first pass at trying to understand how everything is perturbed, how everything is responding.
Thank you. Okay. Oh. So, I'm sorry. Okay. So in the first point of the article is basically to quantify with even much more resolution as it was quantified before, how the different changes in the microenvironment occur in these different models. So for the same, they did experiment in different cancer models in different types of cancer. And the more clear finding in this, what you can see in this stacked bar plot here, is that the different types of cancer models actually show a very different, very clear changes in the fraction of cells that compose each of these tumor microenvironments. Indeed, the most dominant cell type in most of them are the macrophages, and the interesting part of this is that these macrophages are especially affected in these models, MC38 and SV28, but while in the types of melanoma models, or in the models of melanoma, we have more eosinophil infiltration into this tumor microenvironment. But what is even more interesting is that they can do basically all these experiments in which they have a multidimensional data and then use PCA, like a PCA technique that allows you to reduce the dimensionality of all the cells that they were quantifying at the same time. So what we can see in this PCA plot is that what we can see in this PCA plot in the figure in the panel B is that the cells that the different tumor models cluster in different parts of the PCA. But the important part that we have to see is the PC1, which is the one that explains most of the variants in the cells that show more importance, is that most of the tumors, the different samples of different types of cancer models cluster together in the PCA. And more importantly, given that this technique has so much resolution, they can even know in the panel where we see the circle and the arrows, they can even know which cell types contribute more to the definition of these different cancer models. So for example, if we look into the BRAF model, we can see that mostly plasma cells, dendritic cells, and monocytes are the ones that define the position of these BRAF molecules in this particular part of the axis. So this is super interesting because in the microenvironment, we can see which cells have more importance and which cells basically define the signature for for each different model of cancer. Additionally, the other thing that they found is that both melanoma and breast cancer in different types of models of cancer, all of them share common signatures and common features, because when they do this dimensionality reduction analysis, they found that they seem to cluster together, like the variants go together, and also the same cell types go in the same direction in the different models, which doesn't happen, for example, with the pancreatic and colorectal cancer, but happens very clearly with breast cancer and melanoma. And yes, in here, I don't know if, I guess this is a very simplified way of explaining this, but I don't know if the authors would like to add something.
I think that's great.
So, so from these two figures, we can conclude that this technique allows to really differentiate each cell and also determine which populations are the ones that determine the signatures in the microenvironment between the different cancer models. What is also very relevant for the field is that they also did a transcriptional profiling on this at mRNA level, at gene expression changes. And they were also able to find gene expression signatures that would differentiate clearly in the peripheral blood samples exactly which type of cells were coming from breast cancer and which kinds of cells were not coming from a healthy patient. Also, other interesting thing is that these gene signatures were enriched for genes that are relevant for neutrophil and CD8 for T cells and Th1 T cells physiology. So with these two findings, what we can say is that indeed, as it was shown before, and also as the authors show now with much higher resolution, is that the immune system organization is altered across multiple tumor types. And this is very important for what we are going to discuss next. So I don't know if someone from our audience wants to add something or have a question or.
Yes, Kabir, thanks for being here. I have a question and also Bri. So in figure 1, actually, Carlos didn't show, but you also did some analysis looking at the spleen and the infiltrate, and you found that myeloid compartment is somehow enriched, but this is not happening in melanoma. So my question is, I don't know if anyone has done this, but if you, okay, in some of these tumor line models, you put you add or you inject, I'm sorry, the cell line, the tumor cells in and then you will get after some days you will get the tumor. But what if you put like in one side of the mice one type of cancer and in the other side of the mice another type of cancer? What would happen to the microenvironment? So some of the difference that you find associated with one cancer could be like dominant against the other.
That's a really great question. So some tumors, like one of the dominant features that the field has seen in the periphery induced by tumors is the expansion of neutrophils, which is usually driven by the production of GCSF or GM-CSF that is going to stimulate production of neutrophils from the bone marrow. And I feel like wherever you put one of those tumors that produces a lot of this like growth factor, you're going to get massive expansion of neutrophils. And some people have done some studies, and I have done some experiments as well, where I will put one tumor of a different type on one side and then a tumor of a totally different type on another side. And what I was trying and what some other groups I know have tried is to see if one tumor begins an immune response and then you seed another one later, the immune response to the second one that you seed later is different. But whereas if you were to put like one of these melanoma models that seems to have a smaller systemic impact on the spleen compared to one of the breast tumor models that has a much larger impact on the spleen, I'm not really sure what would happen, but that would be interesting. I would guess that the GCSF produced by the breast tumor cell lines would dominate the kind of smaller systemic response that the melanoma models we used had. But it's a really interesting idea.
Thank you.
So now the important question here is that the authors now have reviewed the importance of the different changes in the microenvironment, but now the question is what happens at a systemic level, what happens with the immune system in other tissues that are not in direct interaction with the tumor center. So one of the analyses that they did is this scaffold analysis, in which what you see in here in as little as balls is like the clusters of cells, and the size of the balls determines the size of the cell populations that were found in different tissues. Additionally, the clustering itself refers to not only the I think refers not only to the marker of the cell population itself, but as associated with other features. So if two cells are close to each other, not just at marker level, but also at other features that have been measured for these cells, then the nodes that we see in this network would be closer in this plot. So one of the things that they did is to compare different days of tumor progression, and they compare each day with the day before. And what we are looking at is how the proportion of the different cells is changing across the different time points. So we start with some proportion of CD4 T cells, and these CD4 T cells are gradually diminishing across the different progression of the tumor. And the amazing thing is that this is happening in the spleen. It's not happening in the center of the microenvironment of the tumor, which shows that even though these cells are not in direct contact, in overall the immune system functionality is decreased. And this is, for me, it's extremely interesting. At the same time, we can see how other clusters of cells that were initially not very much enriched, they start being enriched in later and much later phases of the development of the tumorigenesis during the spleen, in the spleen. So other important other analysis that they did with this data was to actually cluster all the different types of cells and across all the different tissues. So this is an amazing experiment because, again, they did mass cytometry in different tissues during the progression of the tumor in this cancer model, and they assessed this in spleen, blood, lymph nodes, bone marrow, and the tumor itself. And it's amazing because with this clustering, we can see the different profiles of different types of cells, and we can see how they are dynamically changing across the progression of the tumor. So one thing that is extremely important, for example, is that the CD8 cluster, it has a substantial reduction in the tumor. It remains not as much as affected in the tumor. No, sorry, it has a substantial significant reduction in the tumor sample, but in the other one, it has an increase, which we can also see in these plots that resemble more the scatter plots from the scatter plots in which we compare CD44 positive cells and CD90 positive cells. We see that in the early days of the tumor, these cells are reducing like gradually from the beginning of the tumor model, but reducing in later stages. But in the case of the lymph nodes, we have the completely opposite trend. So this is amazing because we are talking about the exact same cell type that in the context of the microenvironment, the cell type diminish, but in the context of the systemically in the lymph node, these are increasing. They found similar patterns, but not as strong. Well, yeah, they found good patterns also the same way in the spleen in the cluster 19 that we are seeing in this plotting here. And we also see a reduction of these CD4 T cells, and the same happened in blood. We have in the blood sample, we have an increase in this population. So I think this also shows the relevance of this of using this model, because then we can see how the progression of the tumor growth really changes, not at microenvironment level, but it has tremendous effect in the macro environment. So, and especially in other organs. I don't know if Kenny or someone wants to add something related here with other cell types that you know more detail.
Yes, Carlos, I also wanted to ask, because I didn't know about this. I never heard about eosinophils the role in tumor development. So I don't know if, because in figure 2 you show that there's somehow kinetics of eosinophils distribution within the tumor. So I don't know if eosinophils have like a positive or negative role for tumor development. So what would happen if you do like like a depletion of eosinophils systemically? What would happen to the tumor, at least in this case? And my question is also, why in some experiments you use one cell line or one tumor model, and any other experiment you use other tumor models? That was just my question.
Bree is not able to talk right now, but she's telling me that she sees eosinophils in tumors that are actually responding to immunotherapy. And also eosinophils are important for wound healing. And some people think of a tumor as a wound that can't heal itself. But generally, actually, we thought it was super weird that we saw a ton of eosinophils in the BRAF P10 melanoma model. And if you go back, like, I think even 100, 150 years, one of the first features in the peripheral blood that scientists saw in patients that had cancer was increased eosinophils. And it's still really unclear what role eosinophils are actually playing. they're really harder to genetically manipulate than T cells or macrophages. Also, they're a little bit harder to deplete. So experimentally, they're harder to work with. But I think there was just a review in like the last few days. I think in Nature Reviews Cancer, I'm not sure. But there was a new review about the potential role of eosinophils in cancer that I could maybe like find again and send. They are super interesting, but way understudied. And the other question was, oh, why some tumor models in some parts of the paper? So at first, we wanted to just define the global immune landscape in basically as many tumor models as we could get our hands on. Some of them are widely used in the field, but then we found that the breast tumor models in particular had the most prominent peripheral changes. The mice all get huge spleens. The spleens like triple in size. And also breast cancer in humans and in mice isn't very responsive to immunotherapy. So we thought also maybe this bizarre, like massive changes in the periphery might have something to do with how these breast tumors don't really respond well to immunotherapy. So that's why we started moving on with mostly breast cancer for all of our functional studies.
Okay, and one more question. I got it when I was like reading the paper. Like you showed like with the marker CD101 and CD38 that you have like this population that is somehow associated with non-functionality. So this phenotype is, so it seems that this phenotype is acquired in the tumor, but I don't know if you took these peripheral CD8 T cells and you, I don't know, like do some in vitro assays or looking at to see like their functionality. I don't know if that would work, or if you look more specifically at, I don't know, like epigenetic marks that, like in this systemic context of tumor microenvironments, the epigenetic landscape will be changed. So that will like somehow promote not functionality in the tumor.
Yeah, so those two markers were identified by Andrea Schietinger at Sloan Kettering a couple of years ago, and they represent like the most dysfunctional state that seems to be irreversible. And since in some of our next studies that aren't shown yet with like the challenges of the pathogens in the periphery, we wanted to make sure that we weren't seeing potentially dysfunctional cells in the periphery. We did also do some like ex vivo stimulations to see if they could produce like interferon gamma, TNF alpha, and IL-2 to make sure that cells in the periphery were polyfunctional. But we haven't done any sequencing or epigenetic studies in the periphery yet on T cells. And I think that would be really interesting. So instead, we ended up focusing on just simple in vivo functional studies. But the idea that T cells, even before they enter the tumor, could be dysfunctional, that would be very provocative if that was true. And we only looked at some basic functionality so far.
Thank you so much, Kamir.
I also have a question regarding the interpretation of this, because the technique that you use, you can multiplex several markers, right? And I guess some of those markers will tell you the identity of the cell, but do you have other additional markers that can tell you like dynamic states, like, I don't know, cell death markers or some signaling markers also additional, so you can complement this to your clustering or kind of find more features?
Yeah, in this paper, we used two markers for functional states, and that's Ki67 to mark proliferating cells and
Yes.
cleaved caspase 3 to mark dying cells. And we generally excluded Ki67 and cleaved caspase 3 from clustering because it doesn't define a cell type, rather a cell state. But in the supplement, we do show like shifts in proliferation, for example, across cell types. Yeah, but yeah.
Okay. So far we have discussed the role of the microenvironment and during tumor models, and we also have seen the role of the changes in the microenvironment and the immune system in these cancer models. But now it's very important for the authors to know what happens with the physiology, what happens in conditions in which the immune system have to give a particular response to the environment. So one of the experiments that they did in the model of 83 mice tumor model, what they did is basically to put the mice into a condition of infection, and especially innate immune response type 1. And basically what they wanted to see is that if we have changes in the overall T cell response, that if this would also be reflected in the way the organism responds to infection. So at first glance, just by looking into the phenotype of body weight, what we can see is that even in this model, the uninfected and the infected mice shows the same pattern as the infected control mice, meaning that the way the cells respond to infection doesn't seem to be affected, at least by this particular measurement. The other measurements that they did is to measure the spleen burden of listeria, and they have the exact same phenotype as control with listeria, and also in the model of 83. The interesting part is that the proliferation, when they assess the proliferation using the marker Ki67, what they found is that in this case, the model, which is the infected model, in this case, it shows a reduction in cells in the proliferation of CD8 T cells, meaning that the response is not affected, but the maintenance possibly of this response would be affected in later stages because we don't have any more proliferation, or if you also use as a readout the granzyme B, it would have substantial effects in how these cells can have a cytotoxic activity or not in the same cancer model. So what these results show is that the impairment of T cells in the whole system would indeed not affect the response to infection, but this will be impaired, but in this subset in the context of proliferating cells, and also in the context of how well the cells will respond to the infection, especially in the cytotoxic subset of the cells. So Eugenio, here you wanted to add something?
Yes, so I would like to hear, I know, Brina or Camille point of view, because it's really, particularly this next figure and this one, I really found really interesting, because you found that you have less effector functions, but at the end you have like the same control of the bacteria. Like when we look at the bacterial burden, or we see like it's the same. It doesn't matter if you have being with the tumor or not. So how would you interpret that? I mean, I was thinking that maybe because you have an increase of neutrophils, the neutrophils will in fact impact with the control of the bacterial burden, or I don't know how you interpret this data to have less bacterial, even in this condition, even if you don't have less activating cell of the T cells.
Yeah, so ultimately, this experiment is a test of a new immune response, and it seems that you need a new immune response to control a tumor. And controlling the pathogen is a much easier thing for the immune system to control than another tumor. So you actually need the immune response to listeria and LCMV is far stronger than necessary. It's super easy for the mice to clear it. So there might be some differences like you're saying, the expansion of neutrophils, I have done some experiments to see if like the tumor-bearing neutrophils are phagocytosing more listeria, for example. And in the context of tumors, you get a ton of immature neutrophils as well as mature neutrophils. And it seems that the immature neutrophils are not as good at phagocytosing listeria. So I don't really know more about the cellular response, about what might be happening earlier in the innate response. But ultimately, I think my main point is that this is a much easier thing for the immune system to deal with. And we ultimately just want to show that there are pieces of the immune response that are impaired when a mouse already has cancer, which would extend to them not being able to properly control their own tumor.
Yes, well, thank you. It's really, I really found interesting this part of the paper. So I will be helping Carlos with this figure. If Camille or Bree could also help us, it will be really nice. So the next question. So now they all this experiment in the mice that had the tumor. So now the question is if there's something like intrinsic to the cell. So what they did now, so have these OT1 animals which has all their T cells have a specificity to OVA. They developed in those mice the tumor, and then they purified the T cells and they put it in a wild type mice, and later infected with listeria. Yes, if I'm right. Okay, yeah. So the main question first if to see if there was a difference in proliferation. So now we need to think that in this case we are taking out the T cells that were exposed to this microenvironment, and then now they put it into a wild type mice that don't have this pre-exposure to this tumor microenvironment, and then we see what happens. So in figure A, we see that there is not affection in proliferation at all. So it somehow these T cells are controlling, are responding, proliferating the same. And when they treat the animals with, when we look at, I'm sorry. When in C we see the absolute number, we don't see difference within them. But I think in B, they're looking at the Tibet expression and PD-1 expression of those T cells. So these now are affected by. Just hold on a second. Can you help me with this figure? Come here in B.
Yeah, in B, yeah. So in B, we're looking at OT1 T cells only, and after the cells have completely expanded, so 6 days post-infection. And in the first part, it's just the total count of all of the cells. And so since in the gray bar is the control host, you're able to see more proliferation or more expansion of the T cells when they're placed into the control mouse. But then if you look even deeper at the phenotype of those cells, Tibet is not only associated with CD4 differentiation, but also CD8 T cell differentiation and the acquisition of cytotoxic effector function. And there's less Tibet expression in those T cells that are trying to expand in the AT3 tumor burdened host. And then PD-1 often used as a marker of dysfunctional T cells. So we basically see the opposite. The thing we want to see turning on in cells and the thing we don't want to see turning on in cells. So basically two wrong things are happening with the T cells when they're in the tumor burdened host.
So it's like something intrinsic to the cell?
We think it's not the cells, because the cells themselves are okay when they're in the control mouse, even though we can take them from a tumor. So we now think it's due to the signals that they're receiving earlier in the course of infection from dendritic cells, rather than T cells themselves being impaired.
Yes, thank you, Camille.
Yeah.
So I have just one more question regarding this figure. in the experimental setup. So my question is why you didn't do this experiment? Like I'm thinking about if you could take out, if you do the same model of OT1, but now they put ova melanoma B16 ova. So you have this OT1 with this B16 ova, and then purify the T cells that are systemically, and then put it into a wild type, and now do the listeria. So yeah, because they're like, you're taking out OT1 cells from mice that are exposed to just the microenvironment, but never to the antigen. So what would happen if you take up these OT1s from a B16 ova tumor burden?
So maybe that would be an interesting system. So I'm going to answer it by discussing a like a paper that came out a year ago in Nature Medicine from Ansuman Satpathy and Katie Yost. And they found that patients that were treated, human patients that were treated with PD-1, they looked at the TCRs that were actually inside the tumor before and after immunotherapy. And what they saw is that there was a complete replacement of the TCRs that were fighting the tumor. So this suggests that rather than immunotherapy reinvigorating existing clonal T cells expressing the same TCR that are fighting off a tumor, you're actually completely replacing that pool with new T cells that haven't previously been experiencing the tumor for some reason. So in this case, we were trying to model a de novo immune response where these T cells hadn't actually come into contact with the tumor yet. I think with chronic antigen exposure, we would probably see, like you're suggesting in a B16 ova model, maybe if we took T cells from the spleen, they wouldn't have had direct contact with antigen for so long, so maybe they wouldn't be terminally dysfunctional. But if we took OT1 T cells from the tumors or maybe even lymph nodes of B16 ova bearing mice, they probably would show signs of intrinsic dysfunction due to chronic antigen stimulation. But based on some new papers, it seems that actually an immune response against cancer is from brand new T cells. So that's what we're trying to model here.
Yeah, that I found really interesting. So just before Carlos, we go. So I just want to add some of the results that are actually really interesting in this figure. So I don't think Carlos has it in their PowerPoint slides, but just at the end they really show that there was an intrinsic, there was a defect in the functional state of the dendritic cell. So when we look at markers such as CD80, CD86, and CD83, we see that after infection with Listeria, the animals with the tumor burden have less expression of these markers. And what's really interesting that if they boost the APCs with anti-CD40, you will see an enrichment of CD86, and that will lead to eventually to now reverting the phenotype. So there will be more proliferation of CD8 T cells in the boosted animal in comparison with not boosted. So yes.
Thank you. So the next question to ask in this paper was to see if whether all these changes that we see in the microenvironment after the tumor experience can be reversible, basically. And this actually is very relevant for in cancer, because once you have chirurgically removed a tumor, what happens with the immune system when in this particular situation, or what happens once the tumor is the cancer is under recession. So one experiment that they did is to basically chirurgically remove these tumors and then basically classify these mice into those that start behaving more similar to wild type, that show a more healthier behavior, and start running the same experiments with these mice after the tumor was removed. And one of the interesting things that they found when comparing to the control mice, comparing to the control mice, is that once you remove the tumor in this model, you don't have any more differences in the cell type. So basically, you in the different enrichment of the different cell populations, which means that after you remove the tumor, and they have more or less a systemic response in this sense, that once you remove the tumor, the cells start to going back to their wild type organization. And they can see the same results in different tumor models, that after removing the tumor, you see the how the immune system goes back to normal gradually. If they measure this with a metric of distance, the distance when comparing all the markers of different cells, all the proportions of different cells, what's the distance between the control and the tumor sample? What they found is that, for instance, in the control condition, in the common comparison that we saw from the beginning of the paper, the tumor and the control sample is very far away from the control. But once you remove chirurgically the tumor, you have this sample behaves much more similar to the control sample. They also have cases in which there's local recurrence, that even after you remove the tumor starts regrowing again in some other subjects. And what they see is that in this case, the microenvironment of the immune system, specifically in the spleen, pre-emerge, it have still the same signatures as if you had the tumor there, meaning that there's something there that is shaping the immune system. And once you have, in cases in which you have recurrence of the tumor, the same signatures maintain as if the tumor was still there. Interestingly, they can also find the same results they can find very similar results in the 41 mice. And again, you see how the resected mice have a similar features as the wild type control. But in this case, in the long metastasis, the distance to the control is also closer. And again, the same happens with the MC38 mice, in which the distance between the tumor sample and the effects on the microenvironment are still there after are reverted to control after the tumor is chirurgically removed. So in here, Eugenio wanted to explain this figure.
Here, sorry. In F, where we're looking at the full change of proliferating T cells after the rejection of the chirurgically take out the tumor, and then after then correct me if I'm wrong, Camille, but then after you do the challenge with Listeria, right?
Yeah.
So yeah, so what is important is that when you don't have the tumor, you see that you will somehow reach the same frequency of cells proliferating. We can see clearly in the 61.3% in the animal that has now not the tumor. And yeah, that was in F. And I think in G, we're looking now at the Granzyme expression. So somehow it's rescue here. So the effector function of T cells after taking out the tumor is somehow reached to a basal level, which somehow indicates that actually the microenvironment somehow are affecting the immune response, at least in the case of CD8 T cells. And now, Carlos, I think this figure was really somehow interesting, because they were now trying to dissect which are the molecules or the cytokines or the signaling that were somehow controlling this phenotype. And what they did was using monoclonal antibodies to block IL-1 or GCSF or TGF beta to see if they see this the distribution of immune cells after using these blocking antibodies. And we see that I think they found really interesting data with the GCSF, which was expected. Given GSSF, you will see a reduction of neutrophils. And also with the IL-1 beta, we see reduction of CD8 CD11B cells, but not with TGF-beta. No? And yeah, I think, yeah, and yes, I think that's all. I don't know if Camille want to add something else.
Okay, so I think the paper was really convinced us that about these major discoveries is that one thing is that the response of the T cells is really impaired in the microenvironment, not just in the direct interaction with the tumor, and this effect is particularly clear in the proliferation and differentiation of these cells. Additionally, they found something that was not shown before, is that the type 1 immune responses are fundamental and were previously unappreciated, as this could be very relevant for immunotherapies. And actually, the discovery that I like the most is what the plasticity of the immune system, especially once they were able to remove these tumors, how the systemic basal function of the immune system could be partially put it back. And I think this is extremely important for the design of immunotherapies, because once a patient does not have the tumor, once the patient has the tumor chirurgically removed, then the immunotherapies have to be also adapted to how to the new novel plasticity of the immune system after the removal of the tumor. So I think these are the main findings of the paper, and I think these are very relevant findings for the field. And for our audience, if you want to ask the authors, this is now the time. And thank you.
Thank you, Carlos, for your presentation. Guys, anyone can answer can ask the authors any question? Please raise your hand or write in the chat for your turn.
So I will turn on my camera now. So I will have just two more questions regarding, I don't know if you, because you only focus on CD8 T cells, and then now more and more data indicate that also CD4 T cells are somehow relevant and really important for tumor control. In fact, there are really now new data that indicate that CD4 T cells actually express granzyme B, and they somehow are cytotoxic to the tumor. So I don't know, guys, if you're looking at because in this paper you only focus on CD8 T cells, but I don't know if you're looking now at CD4 T cells too. And my second question is, I don't know if it's physiological relevant or not, but what if you do this, because in the model where you challenge the animal with an infection, in this case with listeria or LCMV, but what if you treat, I don't know if you will see the same, like if you infect with, I don't know, with other bacteria, or I'm thinking about a Th17 response or a Th2, like I don't know, like putting parasite or something to see if systemically the immune response is changed, not only focusing on CD8 T cells.
Yeah, so in these same data generated for this paper with listeria and LCMV, the CD4 T cell response in the spleen is even more impaired than the CD8 T cell response. There are two reasons we didn't include that, which are one is that all of my follow-up experiments to try and see if CD4 T cells were really impaired failed. And another reason is that we were hoping we could use it for a new paper. So currently, people in the lab are following up on CD4 T cells. And the reason my follow-up experiments failed weren't because the phenotype we've seen the phenotype many times. I just managed to fail my experiments. And in the second, your second part of your question, different pathogen challenges. is definitely something I'm trying to convince new grad students that have recently joined the lab to do, since I want to leave. And that would be super interesting. And not only pathogen challenges like type 17 or type 2 immune responses, but also other things like wound healing. Anything that involves an immune response would be really interesting to see how it's altered in not only the tumor models we've already used, but different tumor models. And we do have some evidence that even more types of immune responses are impaired, but we really have skewed towards just the same different flavors of type 1 immune response.
Thank you, Kamir.
Well, now we have a question from the chat. Is from Angel Garcia. Says, in this case, we could consider any kind of influence by the tumor on Treg to suppress even more the responses of CD8 T cells, or even the other of T cells of the leukocytes.
Is this question in reference to the infection models or just generally within the tumor? So we did see interesting Treg dynamics throughout the system over tumor growth. So in the time course data, there's an interesting relationship between the Treg populations in the lymph node versus the tumor, as well as CD8 T cells, where it seems like there's an increase in Tregs in the lymph node that corresponds with kind of plateaus of CD8s in the tumor. So we do think that there's this potentially interactive relationship between the tumor and the periphery in terms of immunosuppression, and that the Tregs within the tumor are not, it's not just those that are important, but also peripherally. But we didn't specifically interrogate T cell behavior or Treg behaviors in the extent of this study, but it would definitely be interesting to look at their influence outside the tumor microenvironment.
Okay, thank you.
Regarding
Yeah,
Gana point, yes, I was thinking about what we were talking before with Kamir. If there's something changing with the Treg, you could also challenge the animal with some autoimmune disease experiments to see if there's a changing of on the Treg function after the rejection of the tumor. So I think it's really interesting, the data regarding Treg and the peripheral change that the tumor could induce on Tregs. That's really a good point there.
Related to that, like some people at UCSF are working on trying to put in mouse models the observation that some people that receive immunotherapy get autoimmune diseases like type 1 diabetes suddenly. So if there are alterations in the periphery induced upon Tregs, maybe we could use that information to somehow either predict or help people that are getting immunotherapy losing their tumors, but then getting a lifelong autoimmune disease, which is, that sucks.
Well, I have questions. Hi guys, I'm Manuel from Mexico, and I want to know if you have plans to analyze maybe the proteome or the transcriptome of the different tumors that you identified in this study, in this work, in order to elucidate what type of cytokines or signals are driving these effects of the tumor on systemic immunologic system?
Yeah, that's a great question. So initially, the way that we narrowed down the cytokines that we perturbed in this paper was we did profiling of circulating factors in mice that either had a tumor, were control, or had resected tumors, with the idea being that if there's a cytokine that the tumor is inducing, that it will be increased in the tumor-bearing animals. It will recede when we resect, because we know the systemic state goes back to a baseline, and then it might come back up with recurrent mice that still have a tumor around. And so IL-1A and GCSF are two that followed that trajectory. And so we perturbed them. We're able to show that they indeed did influence the kind of organization of the systemic state. And we also checked in vitro to show that the tumor cells themselves were capable of producing those two cytokines. So it doesn't limit the obviously contribution of other cell types in vivo to producing them, but we know that the tumor cells can produce them. I think, you know, there's a lot of factors that are going to be produced by the cancer cells. And so that's like, that experimental scheme was a good way to kind of narrow down things that are actually influencing the systemic landscape. We don't necessarily have plans to do a deeper dive into what cancer cells are capable of producing in these different models. But it definitely would be a really great future work to see if there's other important molecules that contribute to this, but also B, figure out in human patient samples what is actually relevant. And there is work that we're doing in human breast cancer patient samples where we're getting blood before surgery and after surgery to see what circulating factors are changed and might be contributing.
Okay, very interesting. Are you planning to knock down or knock out those two cytokines that you have identified?
We tried. It has not worked yet.
Oh, well.
In progress.
Okay, well, we are going to be attending to this data. So Paulina, thank you for your answers. Paulina Lopez asked, do you think that the molecular circadian rhythm in each of these tissues that you studied impacts the dynamics of the immune cells that you observed for each tumor type?
Yeah, so Kamir can jump in on this one as well, but we actually had a rotation student that did an analysis of the systemic immune state over time. So they did time course throughout a day to see how the immune state just changes depending on the time of day. And he did find differences. And so it's definitely a relevant question. For the extent of our studies, we always try to keep it very consistent, like when we were seeing tumors and when we were taking the mice down, so that we kind of control for that circadian rhythm, but definitely think that there's involvement here.
Okay, thank you. Thank you for your answers. And anyone?
Yes, I'm sorry for asking.
No. Please.
So I have just like what I'm thinking now, two more questions that when we think about systemically and regarding the point of the co-stimulation molecules on the dendritic cell. So I don't know if you are, if anyone in the field have looked for the bone marrow composition or the progenitor of this myeloid compartment after the exposure to tumor microenvironment of the tumor burden. So that will eventually lead to a change systemically in all of the hematopoietic cells. And my second question, because I was looking at, and it's always like somehow obvious, you look for changes in spleen or in the lymph nodes, some places that are recirculating the lymph. But now I'm thinking, if anyone have looked for like systemically change in different tissues, like, I don't know, the liver, or all the tissues that are possibly exposed to this, that the tumor burden, in fact, will change the functional state of the hematopoietic cell of the immune cells in all of the tissues of the body.
So the first part of your question, whether or not dendritic cell development could be either functionally or non-functionally altered, is really fascinating, and was part of what I was trying to work on. And then David DiNardo's group at Washington University in St. Louis beat me by a lot. So they came out with a paper in, I think, 2018 in Nature Communications. The first author is Meyer, and it's great. And they show that even in human cancer patients, pancreatic cancer and breast cancer patients, and mouse models, they're able to show that dendritic cell precursor frequencies are significantly decreased. And this is as a result of GCSF signaling through IRF8. So basically, the bone marrow just overproducing neutrophils is leading to a reduction in the production of dendritic cells, which then, at least quantitatively, impairs dendritic cell-based responses. They did not go into function very much. And so that's something we've been really interested in trying to follow up on, is are dendritic cells actually functionally impaired? And if they're functionally impaired, is it at a precursor stage, or is it something that's happening once they're fully developed? And that's something a more junior grad student in the lab is working on.
Regarding this last question, so do you know if, so I don't know if it's the microenvironment, or if you reject the tumor, you will somehow go back to the like the precursor state of the dendritic, like the normal precursor dendritic cell. So yeah, because I'm thinking about this new topic of trained immunity, that after exposure of an inflammatory response, you will somehow, the precursor will have like a phenotype to develop, I don't know, like immune cells with a certain phenotype. So I'm thinking that after exposure to a tumor, even if you don't have your bone marrow or your precursor, will somehow be changed after a while. I don't know if you have looked at that, if there's any related to that.
This was my qualifying exam topic that I didn't end up really getting so far on. We haven't done very much in the HSPC. We haven't done much phenotyping of the HSPC compartment. Generally, when we're looking at bone marrow, we're not enriching for any particular population. So the HSPCs are so few that we can barely detect them. And we're not currently really using markers like CD34, SCA1, anything that can properly delineate them. So, and some of the earliest experiments where Bree was doing the resections, and in my mind, I had this thing of trained immunity. And I was honestly kind of upset that the whole bone marrow went back to normal, because my whole hypothesis was that there was trained immunity and that there would be progenitor cells continually producing neutrophils. And when it went straight back to normal, I was disappointed. That doesn't necessarily negate the hypothesis, because there might still be like epigenetic shifts in like some precursor pool or transcriptomic shifts that maybe upon another insult, they do lead to functionally distinct progeny. I think it's a fascinating idea, and my committee didn't quite agree with me, so I wish you were on my committee, but oh well. I forgot what the second question you asked was.
Yes, like if you, because you look at the spleen and the lymph node changes in the immune cells, but I'm thinking that if there is something systemic, you could also have found changes like in all of the tissues. I don't know, like if like maybe subtle or changes in the immune function of different tissues. I don't know if you have looked.
Not yet. We haven't looked yet. That would be really interesting. Given our lab's expertise in immunology, we would probably look at tissue resident cells, tissue resident immune cells, rather than the organs themselves. There might be some work on this. I feel like in my head there's a paper, but I can't put my finger on it, a recent paper. That would be really interesting, but we haven't looked yet, unfortunately.
Yes, I think I'm sorry.
I was just going to say, I did also check the thymus, and I didn't actually see changes there. Our panel isn't maybe the best to study T cell development, but we didn't see any like dramatic changes in the thymus. So there are cases where it's not having a direct impact.
Yes, I mean, I was looking also, I mean, I was really impressed with the data of the Listeria. I think it's a really good experiment that you could also now like expand this observation with like a second challenge to see if there's a changes in memory T cell formation or something. Yeah, it's congratulations both of you, Brian and Cameron. I really like your paper.
Thanks, and thanks for having us today, guys. This is great. And good walkthrough of the paper. I know it's a lot.
So I don't know if anyone in the audience has any more questions. So I would like to, so we have like this thing that after the talk, if you, Kamir or Brian or both, you could tell the audience like a take-home message for your paper. So what is the take-home message of your paper?
Kamir, you want to take this one?
Sure. So I guess our PI published in Cell, and we're trying to further emphasize his take-home message. After the first paper, we're not sure how much the field paid attention, but it seems like they're starting to pay attention. So his take-home message was literally in the title. It was really clear. Systemic immunity is required for effective cancer immunotherapy. And now we're trying to look at how, so I guess 2 points is that the periphery of immune cell organization is significantly altered, which has functional consequences. That seems better to leave it short, yeah. But we've been trying, my PI has been trying to build on this for a while, and I think it's finally starting to get more appreciated. I guess part of it is his Cell paper was, the figures were even more overwhelming. And this one, we tried to include more classic cellular immunology to maybe have people pay attention a bit more.
So thank you. So just before we leave, we also like to take a picture of all the audience. So I don't know if you guys could turn on your camera. Let me see. Thank you for all our audience to joining us here in Journal Club. So we hope we can see in future sessions. Thank you, Kamir. Thank you, Brian, for your time and your answers. And congratulations for your work. So I want in 1, 2, and okay. 3, we got the picture. Thank you, guys. Thanks, Kamir. Thanks, Brianna.
All right,
Thank
thanks,
you.
guys. Take care.
Thank you.